Dr. Evrim Unsal is our next speaker. She is an embryologist since 1997. She received her PhD in biotechnology, specialized in reproductive genetics. She became an associate professor in 2016, lecturing in medical genetics. As a molecular biologist, she is director of Microgen Genetic Diagnostic Lab, focusing on PGT. Currently, or recently, she was an associate research scientist at Yale University Fertility Clinic from August 2023 to August 2024. And nowadays, she continues her position leading the molecular genetics department at Microgen Genetic Diagnostic Center in Ankara, Turkey. Dr. Unsal, thank you so much for joining us, and please go ahead with your presentation.
Thank you, Berthe. It's my pleasure to be here. And actually, first of all, I'd like to thank Dr. Gilles Palmer for inviting me to participate in this webinar. And I'm glad to have the opportunity to share my work here. And I appreciate everyone who have contributed to organization this event. And my disclaimer for today is like that. And first of all, I wanted to share with you the PGT Good Practice Guideline prepared by the Turkish Society of Reproductive Medicine Genetics Special Interest Group, which I was coordinating this group, and a consortium of 26 experts in the field of PGT. And the summary report focusing on just PGTM session has been published in RBM Online, and all the attendees can also access the full version on the TSRM website. During my presentation, I will explain key points about blastocoel fluid and its role in PGTA. And I'll start by defining what blastocoel fluid is and how it's collected in PGTA procedures. And I'll also highlight the advantages and limitations of using blastocoel fluid for genetic testing. And after that, I'll discuss why PGTA is controversial and how we can improve the efficacy of this test. Since I am the last speaker, some of the content may be repeated, but I'll present it as I have prepared it for the integrity of the presentation. The presence of DNA in blastocoel fluid was first reported in 2013 and opening a new era while discussions about spent culture media were ongoing. And the collection of BF DNA is not completely non-invasive. And the key question remains if this DNA accurately reflects the genetic status of embryos. At that time, discussions were ongoing about the accuracy of the test and the potential detrimental effects of blastomere or trophectoderm biopsy on the embryo. And as you see over here, the reported concordance between T biopsies and the spent culture media showed significant variability, ranging from 33% to 90% almost. So similar variability in reported concordance was also observed in BF DNA. And these discrepancies may be related to differences in different culture protocols and DNA testing methods, potential external or maternal contaminations. While it remains a promising approach for embryonic genetic testing, it's still under investigation and cannot be offered as a diagnostic test until the origin of the DNA is clarified and concordance results are reliable as with at least as with T cells. Today, it's been adopted by some laboratories as a tool for prioritization and defining the competent embryos for the transfer. In a study published by Dr. Magli and her colleagues, blastocoel fluid biopsies were performed and 30% of these samples couldn't amplify successfully. 94% concordance rate was observed between aneuploid status of T cells and BF. However, of this total concordance, 66% was reported as full concordant, and rest is partial concordant. And in this study, overall amplification success rate was 21% across all samples. However, 55% of these haploid embryos failed to amplify DNA, while 19% of the unhaploid embryos revealed amplification failure. According to researchers, these findings can be used for selecting the most viable embryo for transfer, and they speculate that priority would be given to T haploid blastocysts having BF with amplification failure. As you know, the search for markers such as time-lapse prediction, metabolomic testing, morphokinetics to obtain a competent embryo has been going on. And the embryos with non-amplified results would be selected for further TE biopsy, allowing only pre-screened embryos to undergo PGTA. However, considering that even the TE biopsy process itself remains controversial, and when the factors such as cost effectiveness and the potential risks to embryo are considered, I believe this approach may not easily become a part of routine clinical practice. Technically, since spent culture media is a non-invasive method, the semi-invasive blastocoel fluid biopsy has actually only been the focus of limited study groups. And although many studies have looked into SCM samples, no established method for routine clinical use has been introduced yet. And the study I'm referring to actually focused on comparing the effectiveness of SCM and the BF samples by looking at the ploidy concordance of both techniques. And I won't dig into details to save time, but briefly, they found that SCM samples showed more than 5 times higher pre-VGA DNA content compared to BF samples and higher concordance rate with the blastocyst and the inner cell mass. And according to me, since acceptable consistency rates have been achieved, SCM technology isn't quite ready for the routine clinical application. In this context, many research groups suggest that SCM technology could be used as a way to prioritize embryos with higher pregnancy potential. However, because it's difficult to standardize sample source across IVF labs, these methods are not yet ready to become routine in genetic tests. Building on challenges and limitations we have discussed, it's important to note that the ESHRE PGT consortium has reported that these techniques are still in the validation phase, and this means that further optimization is needed before they can be widely adopted. And when we look at the nature of blastocoel fluid, it's like a collection of various apoptotic cell products like proteins, mtDNA, mRNA, and DNA. And these DNA fragments ranging from 160 to 220 base pair in length are very similar in nature to typical apoptotic fragments circulating in the blood. So BF DNA often originates from necrotic or apoptotic cells, and this can affect both quantity and quality of DNA. And this leads to significant variation in amplification rates and concordance with TE biopsies. Differences in technical expertise in performing blastosynthesis is also an issue for the inconsistency. And for these reasons, despite the potential clinical benefits of blastosynthesis, its use is still under investigation and not fully reliable for routine testing in current state. Technical limitations and heterogeneity in technical expertise with blastosynthesis contribute to difficulties in assessing its validity. In current methods, all the fluid from blastocoel cavity is aspirated using an ICSI pipette. However, transferring this fluid into the PCR tube without losing any amount is technically challenging. Some practitioners use a very small droplet on the Petri dish during the biopsy, then try to transfer the fluid into the droplet before moving it to the PCR tube. this method can still result in DNA loss during the transfer from droplet to tube. When trying to transfer the fluid directly from the ICSI pipette to the PCR tube, there is a risk of breaking the glass pipettes, which can cause issues that might inhibit PCR reaction. And in order to avoid stiffness of the fluid, a preloaded microneedle with media can be used, and this volume will also provide extra pressure while releasing the fluid into the tube. An additional manipulation and cost burdens, such as needle replacement for each embryo biopsy, limits the methods as well. And actually, the tubing protocol is always an issue in PGTA applications. And to address this, Leo and his colleagues introduced a tool you can see over here, and that allows direct transfer of the biopsy trophectoderm cells into the PCR tube under an inverted microscope during manipulation. And to avoid sample loss during BF transfer, such a tool can also be used to increase DNA yield and improve the accuracy of PGTA testing. With this approach, a more precise concordance study can be performed for BF DNA as well. Despite the ongoing debate about PGTA, as you can see from our NGS-based PGTA embryo referrals, demand has been increasing every year. And this graph shows data from the first quarter of 2024. And of course, like a techniques, PGTA has some advantages and also disadvantages. Thanks to possibility of selecting euploid embryos before transfer, PGTA provides a higher possibility of successful pregnancy and reduction in spontaneous miscarriages. Among many different reasons, PGTA is still controversial, and in particular, nowadays false positive results has become a significant point of debate among IVF practitioners. One of the key issues in these controversies is the retesting of embryos that were initially reported as chaotic. Interestingly, some of these embryos end up showing euploid results after rebiopsy. For example, in this study, 4% of 3,000 embryos tested via PGTA were initially identified as chaotic. When 54 of these embryos were retested with a new biopsy sample, 4 remained chaotic, but 9 were found to be euploid. And similarly, an abstract from Turkey showed that samples reported as chaotic or inconclusive by one laboratory yielded different results when tested at the other laboratories. To assess the controversial outcomes of PGTA, we need to identify the potential sources of these problems. These issues should be assessed by both IVF and genetic perspectives. Unfortunately, since there are only a limited number of reproductive genetic laboratories, these problems are often raised by IVF practitioners in the literature, and the assessments may lack the viewpoint of genetic labs. As a former embryologist with the experience from both perspectives, I'll briefly summarize the situation in the next few slides. First, these evaluations must be evidence-based, and each laboratory should use KPIs and benchmarks. With this approach, my colleague Dr. Bashar from Yale University prepared a review on IVF KPIs, and I contributed to the section of PGT KPIs. The first step always is a physical examination of PCR tubes, followed by whole genome amplification. And the samples are then sequenced using NGS, where MAPD values, productive read counts, and quality scores gives us quantitative way to assess sample quality. When the PCR tubes with the biopsy samples first arrive at the genetic laboratory, the first step is a physical examination. When performing cell tubing on 2 microliters of PBS, adding extra volume can affect the PCR reaction and increase the chances of false positive results. Similarly, during his talk at 2022 PGDIS Congress in Berlin, Dr. Richard Scott mentioned that in their lab, Different embryologists showed variations in their tubing techniques. He pointed out that when tubes with an increased volume, like 3 microliter, were used for NGS, the MAPD values were affected. Since these types of samples are valuable material, genetic laboratories conditionally accept these inappropriate samples after physical examination. The second stage of quality control is the assessment of amplification success after whole genome amplification. This is done by either spectrophotometric measurement or gel imaging, depending on the platform used. And here you see a batch of 96 samples, almost all of which were successfully amplified. However, tubes marked in red indicate amplification failure here, and while those marked in blue indicate weak amplification. MAPD is one of the most important quality control metric used to assess the noise level in next generation sequencing data, and it evaluates the variation between the coverage of the probes across the genome. And lower MAPD score indicates less noise and better quality data, while higher MAPD score suggests more noise and potentially unreliable data, and which can affect the accuracy of PGT tests. Generally, a MAPD score should be below 0.3 is acceptable for high quality data in PGT analysis. Here you can see cytoviews of the cells with different amplification level. And the first image is from a well-amplified sample, and MAPD value is 0.2, and this is a very important KPI to evaluate sample quality. Weakly amplified samples mostly reveals NGS biases. In the second view, gender information is missing, and due to low quality, X and Y chromosome log ratios cannot be accurately reported. The last one is from a sample with amplification failure, and last 4 samples are inconclusive due to insufficient MAPD value and cannot properly interpreted. When we see these bias aneuploidies, doing a second analysis with a new biopsy might actually show euploid results or different results. To keep short, sample quality is the most important factor for getting accurate results with the PGT technique. And the read count is another crucial KPI. For PGT, a read count of over 70,000 is needed to ensure accurate results. As you can see here, samples with low read counts can be reported as inconclusive due to poor data quality. And here there are these highlighted 3 embryos from the same patient were discarded due to ineffective tubing. However, highly reproducible results were achieved in the remaining patients from the same NGS run. It should be acknowledged that the rates of mosaicism, segmental aneuploidies, and inconclusive embryos in PGT testing are operator-dependent procedures. Reference genetic laboratories that provide testing services for many IVF clinics, they run 96 or 192 embryos simultaneously. So we can clearly identify differences among centers and even between embryologists within the same IVF clinic. And the KPIs I mentioned play a crucial role in providing feedback to IVF laboratories. In fact, these evidence-based metrics have recently been reported by leading groups conducting significant research in this field. And this study, the quality assessment of biopsy and tubing techniques by embryologists working in the same labs, were evaluated at the NGS stage, and attention was drawn to the deviation that contribute to NGS biases. Since we have been giving service to IVF laboratories in 42 different countries, we have been experiencing such a different bias and no result ratios among the centers. And these technical errors can be arise during biopsy or tubing stages. And we always give feedback when we see unexpected fluctuations. And as you see in gel electrophoresis result over here, first 7 samples didn't amplify properly. And when we investigate the root cause, we found out that calcium magnesium free media was used for biopsy, which inhibits PCR reaction during VGA. Additionally, optimal washing procedure should be performed in order to avoid such a inhibiting contamination of these inhibiting factors, and vigorous washing should be performed by the embryologist before tubing. The other faulty biopsy approach is to remove leftovers of cell debris instead of TE cells for the intention of minimal invasiveness. These debris are not reflecting the embryo reality and reveal false positive results. Tubing of the biopsy cells has a great impact on PGTA results. And from our experience of working with IVF laboratories that perform different tubing approaches, standardization of tubing method is crucial for robust genetic test results. Inappropriate tubing always reveals inconsistent results. And actually, we shouldn't transfer biopsy cell batch without washing vigorously. We do not perform tubing at eye level, and we should always follow the releasing of the cell into the tube under the stereo microscope. And we shouldn't perform tubing while any of our colleague is making vitrification in the same cabinet, and keep away our tubes from the liquid biopsy. And we should remove cumulus cells efficiently, especially in PGTM cases, and take cautious to avoid external contamination, and transfer biopsy cells with a minimal amount of media, which is very important one. And we should change the tips for each embryos to avoid any contamination as well. And there's a limited studies focusing on inappropriate tubing effect. And for this purpose, we design a multi-center comparison study to evaluate impacts of different methodologies on genetic outcomes. For this purpose, we have been comparing conventional tubing method, which is cells are transferred to PCR tubes containing 2 microliter molecular grade PBS, and the dry tubing method, cells are transferred directly to the bottom of the empty tube, and the droplet method, cells are transferred in the microdroplet made at the bottom of the tube. And the colleagues from Yale University, we conducted embryo biopsy for donor embryos for research aim. And according to preliminary results, with the 48 sample, we obtained successful amplification in all these samples. You may see the initial samples and the gel view of the whole genome amplification products. Among all methods, no major inconcordance was observed, apart from minor differences in mosaicism results, which will be evaluated once full data set is ready. Because we are gonna perform a comparison study for 400 samples, trophectoderm biopsy samples, and we will compare these different types of tubing methodologies. And only one sample we have detected a segmental duplication with a single method, which is considered as a technical artifact. And once the project is finalized, we will publish the efficacy of each methodologies. I'd like to thank to my teammates and the owner of the Microgen, Professor Dr. Baltacı, and thank you for your attention. And if you have any questions or would like to discuss this topic further, feel free to reach out to me and I'd be happy to connect.